Have you ever wondered why some people burn fat efficiently while others struggle to manage their blood sugar, no matter how disciplined their diet seems? The secret lies deep within your cells in a metabolic tug-of-war first described over 6 decades ago. This is the story of Sir Philip Randle and the discovery of one of the most elegant yet misunderstood mechanisms in human physiology, the Randle Cycle, also known as the glucose fatty acid cycle, a biochemical dance that determines whether your body runs on sugar or fat and how that decision shapes your long-term metabolic health. Before we get too deep into this topic, hit that like button and subscribe to the channel. This will help bring the truth about health and healing to the thousands of people who need to hear this message. Now back to the episode. Sir Philip Randle was a British biochemist whose curiosity about energy metabolism led to one of the most influential models in metabolic science. Born in 1926, Randle worked at the University of Cambridge and later the University of Oxford, where his research focused on how cells decide between burning glucose or fatty acids for fuel. In 1963, Randle and his colleagues published a groundbreaking paper describing what they called the glucose fatty acid cycle. Their discovery revealed something revolutionary, that carbohydrate and fat metabolism compete for dominance inside the cell. This insight reshaped how scientists understood insulin resistance, diabetes, and obesity, decades before these conditions became the modern epidemics they are today. At its core, the Randle cycle explains a metabolic principle. When fatty acid oxidation is high, glucose oxidation is suppressed, and vice versa. Your mitochondria, the energy factories of your cells, must choose which substrate to burn for ATP production. When you consume a carbohydrate-rich meal, glucose floods the bloodstream. Insulin rises, glucose enters cells, and fat oxidation decreases. But when your diet is rich in fat and low in carbohydrates, fatty acids enter the mitochondria for oxidation, inhibiting glucose utilization. This metabolic switch isn't inherently good or bad. It's simply a reflection of cellular preference based on availability and hormonal signals. However, in our modern diet, where both fats and carbohydrates are chronically elevated, this system becomes jammed, leading to metabolic dysfunction. To understand this process deeply, we must look at the enzymatic interactions that govern it. Inside the mitochondria, fatty acids are broken down through beta oxidation, generating acetyl-CoA and NADH. When fatty acid oxidation is high, these molecules accumulate, inhibiting pyruvate dehydrogenase, the enzyme responsible for converting pyruvate from glucose into acetyl-CoA. As a result, glucose oxidation slows. At the same time, elevated acetyl-CoA levels stimulate citrate accumulation, which exits the mitochondria and inhibits phosphofructokinase 1, a rate-limiting enzyme of glycolysis. This double inhibition effectively shuts down glucose metabolism when fats are the primary fuel. The reverse is also true. High glucose oxidation suppresses fatty acid entry into the mitochondria through reduced carnitine palmitoyltransferase 1 (CPT1) activity. This reciprocal inhibition forms the elegant metabolic seesaw known as the Randle cycle. Hormones play a decisive role in steering this metabolic balance. Insulin, secreted by the pancreas in response to rising blood glucose, promotes glucose uptake and storage while suppressing lipolysis, effectively turning off fat oxidation. Glucagon, on the other hand, does the opposite, stimulating lipolysis and hepatic ketogenesis when glucose levels fall. Catecholamines, cortisol, and growth hormone further modulate this balance, especially during fasting, exercise, or stress. In a healthy system, these signals maintain metabolic flexibility, the ability to shift between carbohydrate and fat fuel sources efficiently. But in metabolic disease, chronic nutrient overload and hormonal dysregulation lock this switch into one direction, often toward glucose dependency and fat storage. When both glucose and fatty acids are chronically elevated, as seen in high-calorie, ultra-processed diets, the Randle cycle becomes dysregulated. Cells experience substrate competition overload. Fatty acid oxidation inhibits glucose metabolism, yet glucose continues to flood in under insulin's command. This mismatch creates metabolic congestion, mitochondria overwhelmed by simultaneous inputs. Over time, the result is elevated intracellular lipids, reactive oxygen species, and inflammatory signaling that blunts insulin sensitivity. Insulin resistance develops not from too much sugar alone, but from the combination of excess fat and carbohydrate intake. Sir Randle predicted this decades before the obesity and diabetes epidemics emerged, a prophetic insight into the biochemical foundation of modern disease. Understanding the Randle cycle helps explain why dietary interventions such as low-carbohydrate, ketogenic, or intermittent fasting protocols can restore metabolic balance. By lowering insulin and reducing glucose availability, fatty acid oxidation increases, improving mitochondrial efficiency. Conversely, during periods of high carbohydrate intake, temporarily suppressing fat oxidation is normal, as long as the system can switch back. Metabolic inflexibility, not the use of either substrate alone, is the hallmark of dysfunction. Regular exercise, fasting intervals, and nutrient-dense whole foods all enhance the body's ability to toggle between these fuels smoothly. In essence, optimizing your Randle cycle means reclaiming your cell's ability to decide intelligently which fuel to burn. From type 2, diabetes to cardiovascular disease and even neurodegeneration, the fingerprints of Randle cycle dysregulation are everywhere. In the heart, excess fatty acid oxidation impairs glucose utilization, leading to cardiac inefficiency and diabetic cardiomyopathy. In skeletal muscle, chronic fat oversupply reduces glucose uptake, impairing energy metabolism and performance. In the liver, simultaneous exposure to fructose and fatty acids drives non-alcoholic fatty liver disease, NAFLD. And in the brain, insulin resistance linked to poor metabolic flexibility contributes to cognitive decline, sometimes described as type 3 diabetes. Each of these conditions can be traced back to the same fundamental imbalance first mapped by Sir Randle's simple but profound model. Restoring Randle cycle balance requires strategic simplicity. Prioritize unprocessed, nutrient-dense foods. Separate carbohydrate and fat intake when possible. Avoid high-fat, high-sugar combinations. Engage in regular movement to enhance mitochondrial turnover. Incorporate fasting or ketogenic phases to train metabolic switching. Support insulin sensitivity through adequate sleep, stress reduction, and micronutrients like magnesium and chromium. These aren't trends. They are biochemical truths derived from the fundamental design of human metabolism. Sir Philip Randle's work reminds us that the human body is not simply a calorie calculator. It's a dynamic, adaptive system guided by metabolic intelligence. When we understand and honor that design, we move beyond diets and into the realm of cellular wisdom. The Randle cycle is not just a biochemical model, it's a roadmap back to metabolic freedom. If you found this information helpful, please like this video, subscribe to the channel, and leave me a comment. Let me know what medical science topic interests you most. For more information about our clinic, please go to drvictoriamunoz.com or see link in description below. We have helped thousands of people regain their health and vitality, and we can help you. I am Dr. Victoria Muñoz. Until next time, take care.